Hello, I am Nur Atika, and along with my friends Dayang Nur Afika and Aina Nadra, we'll explain about lipolysis and beta oxidation. Have you ever wondered on how your body gain energy while you are fasting? The answer is our body gain energy by the process of lipolysis that is happen in our body. Lipolysis is the breakdown of lipids and involves hydrolysis of triglycerides into glycerol and 3 fatty acids. Where the cytoplasm is the compartment for this process and the nature is catabolic. The rate-limiting enzyme is HSL, which is hormone-sensitive lipase. Hormonal control for lipolysis is stimulated by glucagon, cortisol, and catecholamine, inhibited by insulin. Fatty acid is the product of lipolysis. There are 4 types of fatty acid, which is beta, alpha, omega, and peroxisomal oxidation. In this video, we will explain more on beta oxidation. Beta oxidation is a process through which fatty acids in the form of acyl-CoA are broken down in the mitochondria in order to give energy. The substrate is fatty acid and the product is acetyl-CoA. The site of this process is in liver, lung, heart, adipose tissues, skeletal muscles, and kidney. The compartment of this process is in mitochondria and the nature is catabolic. Dayang Nur Afika and Aina Nadra will explain more on lipolysis and beta oxidation.
Thank you, Nur Atika Halik. I will now focusing on the process known as lipolysis. Lipolysis is a catabolic process leading to the breakdown of fat stores known as triglycerides found within the adipose tissue. Here is a quick illustration of the major components that make up a triglyceride molecule. Let's begin with the glycerol backbone. The yellow one is the 3 fatty acid tails and the 3 ester bonds that link these 3 fatty acid tails to the glycerol backbone. The major components of a triglycerides are the 3 fatty acids, a glycerol backbone, and the 3 ester bonds. As I mentioned earlier, lipolysis involves the breakdown of triglycerides. It releases the attached fatty acids away from the glycerol backbone. In order for this to occur, each of the ester bonds needs to be cleaved by a process termed hydrolysis, which simply means breaking bonds through a chemical reaction with water. The whole process requires 3 separate steps. Each step cleaves one fatty acid away from the glycerol backbone until all the fatty acids are liberated. The first step involves the hydrolysis of the first ester bond. During each ester bond cleavage, the blue hydrogen that bond to the oxygen on glycerol backbone, while the remaining blue HO attaches to the carbon of the fatty acid residue on the right. Here is the illustration of the products of the first step. As you can see, the first step liberates the first fatty acids away from its parent glycerol backbone. That means the original triglyceride has lost one of its fatty acid to become a diglyceride, where 2 number fatty acids attached to the glycerol backbone. The enzyme required for the hydrolysis of this first ester is termed adipose triglyceride lipase. The second step involves the hydrolysis for another ester bond. The step is the same as step 1. Diglycerides has lost one of its fatty acids to become a monoglyceride. The enzyme required for this second step is known as hormone-sensitive lipase. For the third and the final step, where involves the hydrolysis of the middle ester bond. The first step was repeated again for the third and the final step. Once again, the original monoglyceride has lost one and only fatty acid to become the alcohol known as glycerol. The enzyme required for this final step is known as monoglyceride lipase. In summary, during lipolysis, 3 water molecules hydrolyze 3 ester bonds within a triglyceride molecules in the presence of 3 different enzymes to liberate glycerol plus 3 fatty acids. The resulting fatty acids are oxidized by beta oxidation into acetyl coenzyme A, which is used by the Krebs cycle. This part will be presented by Aina Nadra.
Thank you, Dayang Nur Afika. Now I will proceed to beta oxidation part. Beta oxidation, or also known as fatty acid beta oxidation, is a multi-step process by which fatty acids are broken down the long chain of acyl-CoA molecule to acetyl-CoA molecule. The number of acetyl-CoA produced depends upon the carbon length of the fatty acid being oxidized. This process involves a variety of enzymes, with the four main enzymes involved in fatty acid beta oxidation being, in order, acyl-CoA dehydrogenase, anoyl-CoA hydratase, hydroxyl-acyl-CoA dehydrogenase, and ketoacyl-CoA thiolase. At the end of each beta oxidation cycle, two molecules are formed, an acetyl-CoA and acyl-CoA that is 2 carbons shorter. Now, let's look at how a 16-carbon fatty acid palmitoyl-CoA is broken down completely into 8 molecules of acetyl-CoA through the process of beta oxidation. Let's begin with the 16 carbon of fatty acid, which is called palmitoyl-CoA. Here is the alpha and beta carbon of acyl-CoA. Then, we proceed to the process of first reaction that takes place in this beta oxidation series, which is the dehydrogenation reaction, where it removes the hydrogen atom. Hydrogen atoms are removed along the beta and alpha carbon, which cause a formation of a double bond between them. The compound that being produced is called trans-anoyl-CoA. The enzyme that catalyzes this reaction is called acyl-CoA dehydrogenase. You can see where the hydrogen atom is removed from the alpha and beta carbon structure. These hydrogen atoms are donated to the electron acceptor, which is FAD, that be reduced to FADH2. The reason why the molecule is called trans-anoyl-CoA is because of two hydrogen atoms now in trans state. Now we proceed to the second reaction in this beta oxidation, that being called as hydration, which means addition of water. As you can see, a hydroxyl group is added to the beta carbon structure, and meanwhile a hydrogen atom is added at the alpha carbon structure. The compound that being produced is called hydroxyl-acyl-CoA, and the enzyme that be involved is anoyl-CoA hydratase. Next, we proceed to the third reaction involved in this beta oxidation series, which is the dehydrogenation reaction. As you can see, hydrogen is removed from the beta carbon of hydroxyl-acyl-CoA, which causes the formation of double bond between carbon and oxygen atom. Then, the hydrogen that be removed is accepted into the NAD that be reduced to NADH, and this reaction is catalyzed by the enzyme of hydroxyl-acyl-CoA dehydrogenase. The compound that be produced in this reaction is called ketoacyl-CoA. Ketoacyl-CoA is much more symmetric and balanced molecule, which can be broken down into 2 molecules. The last reaction involved in this beta oxidation series is cleavage reaction that we call thiolysis. This reaction uses a molecule of coenzyme A and being catalyzed by the enzyme thiolase. Then the ketoacyl-CoA were broken into 2 molecules, where the first is a new fatty acid which has a side chain of 14 carbon called myristoyl-CoA, and the second molecule is acetyl-CoA. The first 3 reactions involved in this series of beta oxidation is used to prepare the fatty acid to be more balanced and symmetric, where it can be broken down into 2 molecules. Then the myristoyl-CoA that we produce will again enter the beta oxidation process and repeated until it completely utilized to form 8 molecules of acetyl-CoA through the similar reaction. That's all from me. Thank you. Now I'm going to pass back to Natika for summary part.
To summarize this video, product of lipolysis will oxidize to generate ATP and used to produce triglycerides that are stored as energy to our body. While fatty acids undergo beta oxidation, where 2 carbon fragments match from a fatty acid chain with coenzyme A to form acetyl-CoA, which enter the Krebs cycle when reduced coenzymes are produced. The electron transport chain uses the reduced coenzyme to generate ATP. That is all from us. Thank you for watching.